All right, so I want to give you just an overall summary of the first group of organisms that we will be looking at, but they belong to actually two different domains of life. So when we look at the domains of life, right, there are three domains: bacteria, archaea, and eukarya. The way in which they're related to each other, would be like this. Okay, we have the bacteria, the archaea, and the eukarya. So these are the three domains. Okay, so the bacteria and the archaea are both prokaryotic, whereas plants, so I was going to draw a little flower here. Let's see. Okay. Plants, fungi, and animals, and protists, which kind of left off here, but protists are also belong here, are all eukaryotic cells, either single-celled or multicellular organisms. Most of them are multicellular. So animals are all 100% multicellular. Plants are all 100% multicellular. Fungi are mostly multicellular. There are some single-celled, like yeast, right, that we work with in our class. And then protists, mostly single-celled, but then some multicellular. These guys, the prokaryotes, all bacteria and archaea, single-celled organisms. So write that down. All single-celled for both groups. So single-celled, single-cell. Cells can sometimes live in colonies or groups or biofilms or a variety of different sorts of things that can exist in chains like this, like where one cell's attached to another cell, but each one still functions completely on its own as an independent individual cell. So they're not multicellularity means they sometimes then share different jobs, right, for the overall organism and diversify, right, within that organism. So this is what we're going to be focused on right now. Relationship-wise, what you can see here is in this little cladogram, bacteria and archaea, kind of have things in common. But see, here's archaea and eukarya. What we could see is that the archaea are more closely related to the eukarya than the bacteria are. In addition to that, if we were to start assigning traits and look at distances, so a cladogram doesn't see like evolutionary time. That's a phylogenetic tree. It's something else. And we start to look at very specific things that unite one versus the other or make one more related than the other. We would tend to think, oh, these are really close to each other, much more so than either of them are to eukaryotes. Well, there's another lecture on my protobionts and the origins of cells. You can go and look at and see eukaryotes likely came about as a symbiosis, a relationship between bacteria and archaea where they came together to form eukaryotic cells. And it turns out that the eukaryotic cells carry along with them more of the archaea than the bacteria. So actually, the archaea and eukaryotes are more closely related to each other than the archaea even are to bacteria. So it's kind of like these are two different things. And then they came together to form this, but this group and this group are more closely related to each other than these two are to each other. Just again, kind of weird. You'll find if we look at this kind of relationship, for example, a little round cell called a cocci and a rod-shaped cell called a bacilli. Streptococcus here, say in E. coli, for example. You and a tree have more in common than these two cells, which seems insane. You're like, what? It's like these are just like two types of bacterial cells. They're very small. They don't have much complexity, so they have to be like almost the same. I mean, a tree. Well, we're very different, but genetically, you and a tree often can be found to be more closely related than these things to one another. So that's an idea. There's a lot of hidden diversity in here that we just don't see until we look at the molecular genetics of it and then find there's a lot more diversity there. And then we find between organisms like us that on the surface are very different, we look at the molecular genetics of it and find a lot of similarities. And that's why in a class where we study cells and membranes and chemical reactions, it's important because many of those things are the same for all life, for animals, for all different types of animals, for plants, for all different types of plants, between plants and animals. All the same processes, the same glycolysis, the same enzymes, the same membrane structure. All these things are the same. So what I'm going to point out in this particular lecture here are just for these two groups, what do they have that's sort of the same, and what do they have that's different? As more of a general summary, again, for non-science majors than actually science majors. So start off, cells structured with a membrane. Okay, the membrane is phospholipids, right? And so what we have are the typical phospholipid structure in bacteria, similar to our cells. A little bit different. We have cholesterol in our membranes. They don't, so they wouldn't have the cholesterol there. So bacteria cells, no cholesterol in membrane. It's a little sloppy here in the writing because I'm trying to go faster with this. But the phospholipids would be similar, say, to our phospholipids. Archaea, when we look at their membranes, it looks weird, right? My drawing there, it looks very sloppy, but it's actually on purpose. It's this way. What I'm kind of linking up here are two things. One is that when we look at their phospholipids, some of the tails of these phospholipids can be branched like this, like within a single tail can have branches. So that's weird. That doesn't happen in our cells or bacterial cells. So that kind of says, oh, maybe we're closely related to each other. But that's just one characteristic. That when we look at these relationships, many things are being looked at. This is something unique, for Archaea. In addition to that, when we form the bilayer, right, so really you need to have studied the other material in our class, before studying this, what you'll find is that we have no link between one side and the other. There are no actual bonds between this phospholipid and that one, or this one and that one. And they can flip and change sides, and they can move around as part of membrane fluidity. In Archaea, they can be connected actually to one another. The tails can actually connect and bond from side to side. Because of that, even though the origin of it is a structure which would be individual phospholipids that have unique structure, because the phospholipids that can bind, many people refer to this as a monolayer. But that is a very misleading term, right? Because a monolayer would really just be one layer of phospholipids like this. Monolayer. And then if you form, a little micelle, right? Like the little ball with the phospholipids, that's a monolayer. You have two layers of separate phospholipids, that's a bilayer. It's just that what they have here is really, I would not tend to then say, have you use that term. I would say it's a fused bilayer. That's a little more accurate, right? Really. So Archaea, fused phospholipids, say. So that's something kind of unique. So it makes their membrane structure different. So right from the start, cells are defined by this membrane barrier that creates the cell. And between bacteria and Archaea, certain similarities, but also some specific differences. The next thing. Neither one really has organelles inside. Very large bacteria can actually have membrane structures inside, and some Archaea actually can as well. What bacteria can do is, I'll try to keep some color here. So let's say this is the membrane. So that's going to be, one of these. Bacteria, since they don't have structures inside, but they have ions, enzymes, sugars, amino acids, all these sorts of things, what's going to happen with osmosis? Think about that process, right? Water inside, water outside. But if outside is more water than other stuff, right? And inside we have, amino acids and sugars and ions, etc. What's that mean? That means water is going to want to move into these cells, which means the cells are going to swell. What holds together these? Nothing, no bonds. So the increasing pressure could cause it to burst. Bacterial cells then have, or really have to have, another structure outside the cell membrane, and it has many purposes. Okay, so not just one purpose. This is a structure called their cell wall. This one's dying now. So what we're going to find is bacteria will have a cell wall made of certain material, specific material. Archaea can have cell walls made up of similar but different and sometimes a variety of kinds of materials. Plants have cell walls that are made up of cellulose, so a different material. Animals don't have cell walls, okay? So we don't have them at all. So what we're going to find is some organisms, their cells have cell walls, some organisms don't have cell walls. For the organisms who do have a cell wall outside the cell membrane, it's often made of different material. And that's one of the ways we can say the difference between these different organisms. Like, oh, they have cell walls, but this one's made of X material, this one's made of Y material, two different types of material. That makes them different, potentially different types of organisms or groups of organisms, or very distantly related to each other, and so on. So it's one of the things we could use. So this cell wall is made up of stuff called peptidoglycan, right? Or peptidoglycan, but I'll break it down. Peptid, so like peptide, but it's peptido. And then glycan, I think of glucose or glycogen. It's basically sugar and amino acids joined together in a specific way. We're not going into that in this class. I have other lectures that go over that. For some reason you wanted or needed to know that in more detail, there's lots of good information on that, and I have some as well. But what you need to know is the bacteria have peptidoglycan cell wall, what's called. Now, the Archaea don't have this exact structure. They typically have cell walls. That's cell wall. Okay. The cell walls have great variation. They can have material in their cell wall that's almost exactly like peptidoglycan. It's called pseudomurin. The pseudomurin structure looks a lot like, if you were to look at the actual chemistry of it, peptidoglycan with very, very small changes. So it's almost the same, but it's something different. In addition to that, different types of Archaea can have very different types. So they can have more of just a protein shell type cell wall, called an S layer, and that's maybe all they have. They don't have any of the peptidoglycan type material. Bacteria can have an S layer sometimes on top of this, but they always have this peptidoglycan. So this is very consistent in bacteria. Archaea, more variable, like lots of different variations. Okay, fine. Other sorts of differences come, at molecular levels. So while they have no nucleus and they're going to have the circular DNA, so usually a single chromosome would look like this. A single chromosome. Now, in bacteria, all the genetic information, this chromosome codes for something. Okay. So all DNA typically codes for something. Whereas Archaea can have what are called introns. That's an important one because that's something that links them up with eukaryotes, right, genetically. So what's that mean? It means that some places in the DNA could be non-coding. So it's genetic information held within the chromosome, but it doesn't code for any RNA. It's not anything particularly functional. Sometimes they're regulatory, sometimes we don't know what they do, and still something that's being investigated in different organisms. So these non-coding regions that we have, many of them, sometimes some genes of ours have as much as 80% intron. That means 80% of the DNA in some regions of our chromosomes that would then be copied over doesn't actually code for anything that's used, and then it has to be edited all out before you get to like the spliced shorter form that actually does something right, in the cell. Bacteria don't do that. It doesn't happen in bacteria. All their information kind of is used as is, just directly copied. We have all this variation and things that we don't use. Archaea also have that as well. So that makes, again, something that's Archaea and eukaryotes a little more similar genetically, right, because they have this. All right, some other things, right, about them. Archaea tend to be, not all, but many of them can be extremophiles. Now, some bacteria can live in extreme environments. So it's not that they cannot be extremophiles as well. What do I mean by extremophile? I mean extreme temperatures, right? So temperatures where, say, a normal protein denatured at, say, 45 degrees centigrade. So one of your proteins in your body, it's destroyed. It can't work anymore. 45 degrees centigrade. Some of these organisms, live at temperatures 100 degrees centigrade and above, well above that, right? So temperatures that's not Fahrenheit, that's centigrade, right? So temperatures like that doesn't make sense. Like it's like boiling, it's like that's what we use to destroy things and kill stuff, but they live there and they need those temperatures. So they're extremophiles that are not just thermophilic, but they're hyperthermophilic. They need like great high temperatures even to survive. And that's partly where these fused membrane structure comes in, makes them more stable at really high temperatures. We would just fall apart. Our cells, bacterial cells, things would fall apart at those high temperatures. The proteins would be destroyed at those temperatures, but they have unique adaptations. So high temperature and low temperature. So just extremes. It doesn't necessarily mean high. So high and low. So high and low, not necessarily at the same time. So some organisms are more high, some organisms are more low, but extremes. So extreme temperature, extreme salt concentrations, so they can live at. Sugar, like osmotic concentrations, they can live at a whole variety of different ranges, and some are very then extreme. Different types of gas environments where toxic chemicals exist that would kill most all other living cells, but these organisms live in those environments and can survive and thrive. They actually adapted to those specific environments and not others. They might use sulfur instead of oxygen. Right. So there are those organisms that belong to this group. Are methane producers. The organisms that actually produce methane belong to this group. They don't only exist in extreme environments. Some of these exist in our own bodies. And like I said, in cows, right, that we're known to produce a lot of methane. It's archaea that actually live in the digestive tract of the cows that are producing this methane. So they live in animals in our bodies. They live in the environment. They're just in the soil outside. They're not only in crazy wild places. The point is they are also in these extreme places where scientists would feel like there's no life, there can't be life, it's not possible for there to be life. And then you go and you look there and there's life, there's something living there. Like, how is that even possible? And that's kind of what makes this group of Archaea interesting to study, but also difficult to study, because then you have to create these conditions in a lab setting to study them, and you have to keep them at, say, at this very high temperature, which means you have to have an oven at this high temperature. And that means you have to have equipment, glassware, things that are going at those high temperatures. Then the type of media that we use doesn't work at those temperatures. It doesn't become solid, it liquefies at these temperatures, so we can't keep them on a solid media. There's a lot of challenges that then scientists have to working with them, so that means that they are poorly studied in comparison to many other types of organisms, not because they're not interesting or important, but because they're very challenging to work with. So we know more about just their DNA, kind of extracting it and then analyzing it, than we do the organisms themselves and what they do and how they behave and other roles in the environment. That's something people continue to learn and work on. All right, so Archaea are a type of prokaryotic cell, meaning they don't have a nucleus. They have just free DNA, right, in the cytoplasm, circular DNA like the bacteria, but they're not bacteria. Right? They're their own group, a separate domain of life, and they share some characteristics with bacteria and some characteristics with eukaryotes, with us, with plants and other organisms as well. All right. So again, the point of this isn't to go over all the details of this. I have a few other lectures that go into more of the details. And if you need that or are just interested in that, I'll post those for you to view. However, for this particular information, what do you need to take out of this? The next group of organisms after getting the first cells, we go into bacteria. Who are bacteria and what are they? So I have kind of a separate lecture on that. And then Archaea are kind of mixed in there a little bit, and then we go into eukaryotes. So this is sort of an overview and a bridge between those lectures to kind of give you a bigger picture, and then a couple of the details that either might overlap with the other lecture or might be in addition to. So this is the one to kind of maybe start with, and then you could look at the other ones to fill in all the gaps and the details.